Thank you very much, and appreciate the opportunity to present this first talk about I'm going to have to put my other glasses on here so I can see so I can actually see the monitor here. That's what happens when you get a little older. Okay, monitoring and mitigating the global proliferation of cyanobacteria. And I was glad to see actually some presentations on Lake Biwa, which is a success story in terms of how to get over this problem, but many places in the world are having this problem right now of toxic cyanobacterial proliferation. The question is what to do about it, how to develop tools to monitor and assess the situation better. I think many of you know the linkage in terms of human activities to the global proliferation of cyanobacterial blooms that we're having. I think the first two of these links are clear to many of you. The urban agricultural and industrial expansion, which brings with it more nutrients, but also water use and hydrologic modifications that have a huge impact on this. So the way we handle the water, withdraw water, and use water also plays an important role. And then lastly, climate change, as we've all experienced, including warming, hydrologic changes, play an incredibly important role. And one thing we know about cyanobacterial blooms is we've been studying them long enough in many lakes to know that, in fact, the increase in these blooms is real. It's not just better observation. We are facing a situation where cyanobacteria are coming back after 2 billion years to try to take over the world again. So how do we deal with this issue? The other thing I want to emphasize is that this problem is global. It exists in every continent, maybe except for Antarctica. And the other thing I want to emphasize is that it's not just a freshwater problem. We need to deal with this issue of expanding cyanobacterial blooms all the way from the freshwater to marine spectrum. This problem is expanding into estuaries and even coastal systems, as you can see down below in the Baltic Sea in Northern Europe. Who are the players? Who are the main groups of organisms that we need to be concerned about? There are 3 groups. Many of you are familiar with them. I'll just run through them very quickly. We have the coccoid, solitary, or colonial forms like Microcystis. I think everyone knows Microcystis. This is the number one cyanobacterial bloom problem organism. The second group are the filamentous cyanobacteria that have no cellular differentiation. They include organisms like Oscillatoria and Lyngbya. I think many of you are familiar with them. Some of them can fix nitrogen, some of them don't. And then the third group is the filamentous heterocystis group. They have specialized cells in which the process of nitrogen fixation occurs. And tomorrow when I talk about this issue, we'll talk a bit more about nitrogen fixation and why that's important from an ecosystem perspective. But these organisms, there are groups like the bottom group that can bring their own nitrogen into a lake system. So from a management perspective, this greatly complicates how to deal with them. Why are we concerned about it? Many of these organisms are toxic. They produce metabolites that are toxic to everything from the organisms that are supposed to eat them, like zooplankton, to humans. They also lead to low oxygen conditions. When you get a bloom that dies and creates low oxygen conditions in the bottom water, that creates problems with exclusion of habitats, which directly can impact fisheries, but it also impacts biogeochemical cycling in lakes, the nutrient cycling, the internal cycling. Odor and taste problems. Every water plant engineer knows all about this in terms of how to deal with these odor and taste problems, aside from the toxins that also occur. And of course, lastly, there are huge issues with human use of these waters, including recreational use, fishing use, and sustainability of systems that bring in economic benefits everywhere from fisheries to tourism. Lake Biwa is a good example of a lake where this problem has been solved to enhance the tourist industry. But there are many places where tourism is suffering greatly from this problem. Okay, before considering the parameters that we need to consider in terms of monitoring this problem, I wanted to identify, using this conceptual slide, of the interactive factors that are involved in this in cyanobacterial bloom phenomena. We're all very familiar with the nutrient issue, and in that I would include both nitrogen and phosphorus, but the physics of the system are incredibly important too. And that has to deal with residence time, the rate at which fresh water flows into these systems that are impacted by blooms, the transparency of these systems, how deep light can penetrate into the system, and the mixing rates. All those are impacting the ability of cyanobacteria to dominate systems because they are surface bloom formers. So once they get into a system where there's enough nutrients, they dominate the upper water, which shades the lower water and excludes some of the more beneficial organisms, including macrophytes and other phytoplankton. And that also impacts oxygen production in bottom waters. And these organisms have the ability to regulate their own buoyancy. So they can stay up at the surface and dominate these waters. When they need nutrients, they can migrate back down, come back up. They're very smart. Even though we call them primitive organisms, they actually are incredibly smart in figuring out how to dominate a system that has a lot of nutrients coming in. The residence time is very important. That is the rate in which fresh water that enters the system leaves the system, because actually cyanobacteria are fairly slow growers. So in systems where the residence time is increasing due to either water use or droughts or climatic impacts, that will favor cyanobacteria because it allows them to catch up with other organisms in terms of growth rates. So flushing, for example, is extremely important in terms of minimizing the dominance by these organisms as well. And then lastly, food web issues. These organisms are not readily eaten by zooplankton that then get eaten by fish. So there are impediments here to using the food that's actually produced by cyanobacteria up the food chain. And as a result, the carbon and nitrogen and phosphorus that's produced by these organisms recycles in the lake. It doesn't really go anywhere because the fish that are caught are not eating these cyanobacteria, they're eating other organisms in the food web. So there are all these sort of interactive factors that can lead to enhancement of cyanobacterial blooms. So what do we need to monitor in order to assess the susceptibility of these systems to these blooms, and also as we start to monitor nutrients and as we start to reduce them, what do we need to measure in order to look at improvements or not a lack of improvements in these systems? So these three lists here, and I think my talk will be recorded, so you don't need to write everything down here that's in here in terms of details. There are lists here of physical, chemical, and biological factors that are really important to measure. Light, transparency, the depth of the system is important because that involves the mixing, total mixing rate. The attenuation of light is extremely important. Temperature and stratification are really important. These are parameters that can be easily measured. You don't need high-tech instrumentation to measure these kinds of parameters in order to get get those parameters measured. And then freshwater input and flushing rates are really important. Okay, chemical parameters. The soluble forms of nitrogen and phosphorus are incredibly important. Those are real basic kinds of things that we need to measure in these systems and look at the changes that are occurring. Dissolved oxygen, dissolved inorganic carbon, and then we get to more specific forms of organic carbon and nitrogen and phosphorus. And I've starred those as being parameters that are not absolutely essential, but they're very good parameters to have as we measure the condition of the system and whether it's improving or not. Biological parameters, chlorophyll, the basic parameter for measuring algal production and biomass in the system, and then other pigments that we can now measure using instrumentation like high-performance liquid chromatography, the diagnostic pigments for the different phytoplankton groups that the cyanobacteria are competing with. These are measurements that are made in labs. They're fairly sophisticated measurements, but the samples can be collected anywhere in the world and then shipped to a place overnight using actually just refrigerated sampling, nothing fancy, to determine the total community structure of the phytoplankton that they're competing with. Microscopic identification, very basic measurements. You need to have the expertise in terms of personnel that can identify the phytoplankton, but these are not high-tech rocket science kinds of parameters that need to be measured. Then we get into things like characterizing the community using nucleic acids, DNA revolution, which is now telling us not only what species of cyanobacteria are there, but also the other microbes that they're competing with in the system. Those samples can also be collected in remote places. You don't need to have all that equipment on site in order to measure these parameters. They can be measured in a more sophisticated lab and sent to a lab. Now, I've done a lot of this work out of a suitcase in places like South America, Africa. We're now working in China. We can collect all these samples without having the fancy instrumentation there to actually measure the contents of these parameters. So using shipping and overnight ways to get them to a more sophisticated lab is the way to do that. And then, of course, the zooplankton and benthic fauna. Other parameters, toxins. The toxins that are produced can also be collected in the field and then sent to laboratories to do more sophisticated analysis. Frequency and spatial coverage. When, where, and over what range of time scales and physical scales. And you can see that what I've outlined here is temporally, monthly during non-bloom seasons, weekly to bi-weekly during the bloom seasons when these occur. And if there's a health or ecosystem issue, like fish kills or human maladies, daily monitoring is probably the way to go. Spatially, coverage depends on the bloom location, proliferation, where and when the blooms occur in a system, and then of course environmental health impacts. Well, how does it affect intakes for water supply operations, for example? And we need to locate these sampling locations in terms of major inputs of nutrients that are causing the problem to begin with. One thing that's really important is to now make, I'm going to go down to the bottom part here, is to make, oops, what am I doing here? Was to link these parameters to remote sensing. We now have satellites that are everywhere to be able to link issues like chlorophyll, which gives us an idea of biomass, turbidity of the water column, to remote sensing capabilities. So we should all look for potential links to remote sensing to make these links. Toxins that are produced. This is the whole list of toxins that are now identified. Some of them are actually on the WHO list in terms of particularly problematic toxins like the microcystins, for example. Those toxins can now be analyzed remotely by laboratories by just simply sending samples to these laboratories. And I've just put two detection and analysis systems, including ELISA, the enzyme immunoabsorbent assays. Those are all conducted in the laboratory on samples that can be easily collected in the field and then sent to laboratories, and high-performance liquid chromatography. The upper one is much more economically feasible. The bottom one is much more expensive. But if you know you have a toxin problem or a toxin issue, then you just shift to the one that is more specific, which is the more expensive one, HPLC. But you don't need to use HPLC to begin with. You can start with the ELISA just simply to see whether there's a toxin problem there or not. Okay, one can make use of relatively simple parameters like cell counts to estimate toxicity of these organisms. And I just want to show you a regression that Tim Otten, former PhD student, and I put together of total microcystins, the main toxin that we need to be concerned about, and linking that to actual cell counts of the two organisms that are responsible for microcystin production in most cases, Microcystis and Planktothrix. You can see there's a lot of scatter there, but there is a general relationship there, general positive relationship there between the cell counts of these organisms and whether or not you're exceeding the drinking water standard, the WHO drinking water standard, here 1 microgram per liter, or a recreational standard, whether you're exceeding the ability of someone to actually go in a system and use it for recreational purposes. So again, this can be based on fairly simple parameters measured in the field. And in Lake Taihu in China, where we've been working, we've actually established a very good relationship between chlorophyll A, the simple measurement of the pigments in the algae, and actual microcystin levels in this lake. That's largely because this lake is dominated by a single genus, Microcystis, which is the main toxin producer. So you don't need to go out and do the sophisticated toxin measurements, and that's particularly useful for like drinking water operators who are concerned about whether or not they're exceeding or meeting the safety standards for using the lake for drinking water. Okay, so then once you have use these parameters to actually establish whether or not you have a problem. Go back to the other glasses here. One can do a decision tree analysis, a yes-no type of analysis to see whether you have a problem, whether there's a visible green or scum present. If the answer is yes, then you issue an advisory and start looking for whether or not the negative properties of these blooms are present, particularly toxins, toxigenic cyanobacteria. That can be determined by these simple toxin analyses. If you have that problem, then you go further down the list and you actually start to identify what the organisms are that are producing the toxins, so you can get a better idea of quantitative relationships between the actual player and toxin production. You can go the molecular route or the analytical route. This is the molecular route looking for the genes. You get to the same, essentially get to the same point here using either direct immunoanalyses or genetic analyses. That tells you yes or no whether or not you have the problem, and then look at the specific organisms. You issue an advisory for those organisms, and then your management strategy should then ultimately focus on that particular organism that you have in play. Okay, the last thing I want to mention here is mitigation, because it is in the title of my talk. Where are we in terms of mitigating these blooms now when we have this problem? And you can see there's a list here of different mitigation strategies that have been used to try to control these blooms in aquatic ecosystems. And I've put the first one and the most important one up on top, which is we need to deal with the nutrient issue. No matter what kind of other strategies we impose in these systems, and here they all are, increasing flow to increase the flushing rate in the system, enhancing vertical mixing, which is done mechanically in many systems. For example, in fish ponds that have this problem, they all have vertical mixing capabilities. Sonic disruption, which is now being used as a technique to basically lyse the cells by sonic disruption. Immobilizing the sediments, capping the sediments so that they trap the nutrients and don't release them. That has limited utility, and we'll talk some more about that tomorrow. Dredging, actually removing sediments from the system to remove the nutrients. I'm not saying that these are all good or bad, but these have all been strategies that have been used. Selective grazing and biomanipulation, which is pretty much experimental. And then if all else fails, and you have a small system and you need to get rid of these organisms, chemical application. The chemical application is kind of a last resort because it doesn't really solve the major problem. It just gets rid of it for a little bit of time. Everything still stays in the system. The nutrients still stay in the system. The organisms themselves can go back to the sediments and come back next year when you don't have the algicide in there again. So these are really all kind of temporary fixes for the problem. The real permanent fix is right there. Nutrient reductions. And I'll talk some more about this tomorrow. But any of these physical chemical techniques that are being used, they're all short-term fixes but they don't solve the long-term problem. The long-term problem is to manage our nutrients. And when I talk about managing nutrients, I'm not just talking about phosphorus. We're now in an era where we have to deal with nitrogen too. So this has become more complicated because going after nitrogen is more complicated than going after phosphorus. But we've changed the world tremendously. 50 years ago, you could say, yeah, it looks like phosphorus. If we reduce phosphorus in our detergents, for example, and wastewater, we're going to make a lot of progress. And B was a good example of that, actually, that happened. But now we're putting so much nitrogen in the biosphere through fertilizers, atmospheric pollution, groundwater contamination, that the expenses and the extent to which we need to deal with this nutrient have all increased. We cannot escape the nitrogen problem as well as the phosphorus problem. And I just want to emphasize that. Okay, so very last set of slides. What's being done in Lake Taihu in China, which is this lake that now has 9 months of these blooms to deal with. I will again talk more about Taihu tomorrow. But I just want to give you some idea of what's being done on the ecosystem scale in a system that has these tremendous cyanobacterial problems. Mostly microcystis that's excreting toxins or actually has toxins in it. And this lake, Taihu, it's the 3rd largest lake in China. About 20 million people depend on the lake for drinking water. The lake has changed from a diatom-dominated lake, very desirable, to a cyanobacterial lake within 2 decades, basically. And now we're faced with the problem of essentially putting these mitigation strategies into play based on what we know. So the first thing that has happened in the basin in Taihu is to improve the wastewater treatment. And there's 2 cities on the lake, actually, here, Wuxi and Suzhou. Each one of them have 6 million people in it. Their wastewater is centralized, but up till a few years ago was not treated. So now the big effort is to treat the wastewater for phosphorus and also start treating for nitrogen. And about half the nutrient loads to this lake are actually coming from those 2 wastewater treatment plants. So if we can get at those nutrients and get them out, we're going to improve water quality pretty quickly. And this is essentially part of the big plan for Taihu in terms of reducing nutrient inputs. Using ecological methods to retain the nutrients, particularly from stormwater, but also from agricultural sources, using buffer zones is a strategy that's being used in the Taihu Basin. Construction of wetlands around the lake. You can see that there are different places where wetlands can be established around the lake. The problem in Taihu is that it's 2,300 square kilometers. And we're talking about a very narrow strip around the lake that's only a very small amount of land to use wetlands. So wetlands are not going to solve the problem by themselves. They will help, but they're not going to be the ultimate be-all, save-all in terms of the nutrient inputs. Okay, water diversions. And I think this not only applies to China, but also many other places. You can see here the Yangtze River flowing into the South China Sea. This is Shanghai, Wuxi, Suzhou, and here's Taihu. So the engineer, when this problem occurred back in the, starting in the late 80s and early 90s, the engineers had an answer, and that is, let's flush the lake and build a canal to connect the Yangtze River to Taihu and reduce the residence time of the lake to allow the cyanobacteria not to be so competitive. So this went into place in the late 90s, and you can see here the connection from the Yangtze going, and then here's the exit going into Shanghai. They also pump water back on land in order to do land applications.
5 minutes.
Okay. Yeah, I think I can finish in 5 minutes. So this was the engineer's answer to the problem. Was move the water through the lake more quickly, flush the algae out because we know that cyanobacteria grow slower than the other algae. But there are problems with this. And I wish that 15 years ago the effort would have been a combined engineering ecosystem ecological approach, because the ecological approach was not taken at the same time as the engineering approach. And the problem is that it sounded all really good, you know, from a hydrologic perspective. Take the Yangtze River water, flush it through the lake, force the algae out of the lake, and reduce the residence time. And this is a model, this is a hydrologic model, basically showing you how that impacted the water column itself. The problem, though, is that the Yangtze, these bays here, in which the problem is most extreme, were pretty much isolated from where the water was being forced through the lake. So there was a hydrologic problem there. The other problem was that they were only able to reduce the residence time from about a year to 220 days. And that's plenty of time for the algae to grow many generations. So they just physically could not reduce the residence time enough to flush the algae out. And perhaps the worst problem is that the Yangtze River itself is very polluted with nutrients. So you're actually putting more nutrients into the lake, trying to actually clean up the problem. So if we had included the chemists and the ecologists together with the engineers, we would have had probably a more comprehensive plan. As it is now, about $2 billion has been spent on this engineering effort, and it really hasn't changed the situation at all. In fact, it's made it worse because there's now more nutrients coming into the lake to solve the problem. The other engineering effort that's been put into place was to use sediment dredging in these bays in which the problem is most extreme. That's a good idea on paper, but if you take sediments out of the lake, you have to put them somewhere outside of the basin, because if you don't put them outside of the basin, ultimately the nutrients will come back into the system. And that was not done. So about $1.5 billion was spent on that. And the problem really hasn't improved at all. The other problem with dredging sediments is that you're releasing other things from the sediments when you're dredging it, like contaminants that were locked up into the sediments, putting them back into the water column. So these big hammer physical efforts, they sound real good, and the engineers have quick answers, but you need to take the ecological and biogeochemical components into consideration when you're doing this as well. A lot of people in China that can be put to work cleaning up the blooms. And in 2007, there was a major bloom that caused the drinking water plants in Taihu to be completely shut down. And so one thing that went on was that a lot of people were hired to collect the bloom, bring it to a place where it was processed and put into pellets, and then converted into methane, which sounds really good. But the problem is that only about 1% of what was produced could be harvested in the lake at any one time. So it's sort of like trying to kill an elephant with a BB gun, It's not going to work very well. So this physical effort is limited in terms of its effectiveness. And lastly, flocculation and precipitation of the blooms. That works in the short term. It sort of hides the problem. But it doesn't really work in the long term, because all you're doing is just taking the nutrients down into sediments, and then ultimately in a shallow lake like Taihu, it'll just come back through sediment resuspension. So my plea here is to have mitigation strategies that involve the ecological consideration of what we're doing, and taking a holistic approach where we're looking at the chemistry and ecology as well as the physical factors that we can manipulate in doing these strategies. And lastly, think about both N and P. Not just P, not just N, but we need to deal with both nutrients now. The world has gotten a lot more complicated than it was 50 years ago in terms of dealing with these issues. So just to finish up, what's most practical across these systems? And I've already bashed the engineers enough, I think. No matter what we do in terms of engineering, you still need to deal with nutrients. Reduce both N and P, and this is becoming a very common story now in many lakes around the world that have this problem. Establish the thresholds at which these blooms can actually be controlled by reduction of N and P inputs. That's not easy, but it needs to be done. And we may need to reduce N and P inputs even more in a warmer world, because as things warm up, cyanobacteria like it hot. The warmer it gets, the better they like it. So we may need to reduce the thresholds even more in a system that is experiencing warming. These reductions need to be done year-round. That's because residence time is quite long in these systems, and the legacy effect of loading lasts way longer than a year. So you can't just control P at one time of the year and at another. It needs to be a year-round strategy. And just to finish up, I want to thank many of the people who I've worked with who have made some of their data and ideas available on all this, including my former PhD student who now has a company who's willing to analyze toxins for anyone. Tim Otten, he's the toxicologist. And this again is Kyoto-U, and nothing ever gets done with funding from just one agency. You know, we get funding from everything ranging from the National Science Foundation to the Chinese Academy of Sciences, and work with a lot of Chinese scientists to help solve this problem. And lastly, don't forget about nitrogen. We know a lot about phosphorus, and we've been taught, you know, in our elementary limnology classes, phosphorus is the problem. But nitrogen is part of the problem now too. So the real challenge here is dealing with nitrogen in addition to phosphorus in our management strategies. And I'll just stop there. Thank you.
Thanks, Hans. So time is very limited. Sorry, one quick question. One quick question. Okay, please.
Thank you very much for just giving us the insight on some bacteria. However, few questions. Number one.
Sorry, one quick question.
I'll be around so you can ask more questions later.
Well, with regard to nutrient reduction strategy, I think that one may work very well with the terrestrial input. Because I think for other lakes, for instance, Lake Victoria in Africa, the theories that are being advanced now is that, as far as nutrients input to the lake, it's not about terrestrial. The major is atmospheric or aerial deposition. But how is it that you're going to reduce the nutrients input that are coming from air or atmospheric?
From air?
Yes.
Yeah, from atmospheric sources.
Yeah.
Yes. Well, atmospheric sources can be really important. In Kyoto Basin, they're about 20% of the nitrogen inputs. That is a very challenging part of the issue, because to control the atmospheric sources in China, for example, there has to be a major, rethinking of energy, because most of those sources are coming from fossil fuel combustion, like coal, which is still the main source of energy in China, and automobiles. Everybody in China wants a car now. So that is a challenge. And so there has to be some prioritization in terms of what you go after first. And my advice would be is go for the low-hanging fruit first. Reduce the applications of fertilizers to only what's needed to grow the crops. And I know farmers always like to put on more, than they need to. This is an issue that exists everywhere. It exists in the US, it exists in developing countries, and in fact in developing countries they add even more fertilizer usually than what's needed. Fertilizer is responsible for about half the nitrogen and phosphorus inputs to many of these lakes. So that's a very attractive target. Best management practices like no-till agriculture as opposed to tilling and then just leaving the land exposed to rainfall is another good approach. Animal industry is becoming a huge issue worldwide. Treating waste that comes from animals should be treated the same way that we treat our own waste. In the state that I live in North Carolina, we have about twice as many pigs as people, yet we don't do anything about treating their waste, whereas the cities are forced to treat their waste. So, pigs eat about 3 times as much as people do per day. They generate about 3 times as much waste. It's not their fault. we need to treat their waste just like we treat our own waste. And if we do, just doing better at fertilizer application and dealing with animal waste in a more comprehensive way in North Carolina will reduce our loads by about 50%. But the atmospheric one is a big challenge. Now in the US and in Europe, you know, we have much stricter controls now on nitrogen emissions, so that problem is starting to get reduced. But in other parts of the world, it's actually increasing, like in China.
Okay, thank you very much.